which you'll learn in my course. It's called a probe. Okay, typically, researchers will take a note, if they're ever trying, this is all part of recombinant DNA, if you're ever trying to figure out what a sequence of DNA is, what you do is you take a known sequence, okay, of DNA that you call a probe, and it's radioactive, so you can see the radioactivity coming from it, and you basically put it with that unknown DNA strand, and then you add some restriction enzymes. Okay, you'll learn in this course that restriction enzymes will cut up that unknown nucleic acid strand at a certain sequence, okay, called a restriction site. Okay, so that known probe that you put in that'll also be cut up by the restriction enzyme. And anytime you cut up these different DNA sequences, whenever they're cut up at the same site, so they can combine at the same site. So this is how you make recombinant DNA. You take different DNAs, cut them up, put them together, and naturally they'll reassociate at the same spot, at the same restriction site. That's how you mix DNA together to create new DNA. Okay, which they do in gene therapy these days. Okay, so that's really all they're doing. They took two known hantaviruses, that you can consider those the probes, mixing them with the nucleic acid from the patient's lung tissue. Okay, hybridization. That's when the probe goes in and attaches to that unknown strand of DNA. So when you put it in a microscope under southern blotting, which you'll learn later, you're going to be able to visualize that. And you'll know that unknown strand of DNA because you see the radioactivity emitted. Okay, does that make sense? Okay. Hybridization between nucleic acid strands occur when the base pair with each other. Okay. Hybridization occurred between the synthesized genes and nucleic acid from the patient's lung tissue. Okay? So that means that the DNA they found in the lung tissue matched up, which probably means that there is hantavirus in the lungs if they were matching up, right? Okay? So that's what I'm thinking. But even though the hantavirus is in the lungs, because it's not contagious, it's probably not expelled in the fluid that you cough up or you sneeze out. Okay, it's just in the lung tissue. Viruses will stay in your cells and remain there. And there's two stages every virus go to: a lysogenic phase, a lytic phase. The lysogenic phase is the really mild. It's just where the virus replicates over and over again. The lytic phase is when after all those viruses incorporate its DNA into the host cell, then it starts replicating new viruses called virions, which I think of as baby viruses. Once those explode out of the cell, that's called a lytic infection. That's the really dangerous infection. Okay, so that's probably why this disease isn't contagious, because these patients the virus hasn't entered the lytic phase. It's only in the lysogenic phase. Okay, these are all fundamentals about viruses. It's nothing too specific. Okay, and then finally, and then another thing I always think about a virus is, most of the time we can't see viruses with a light microscope. Okay, viruses are the smallest things, one of the smallest things we know. Okay, so we can't see them with microscopes. We can see bacteria with microscopes, but viruses are way, way, way smaller. So we have to have an electron microscope to view them. Okay, and then finally, experiment 3, genes from the new pathogen were inserted into bacteria to produce viral proteins. Okay, and that is a process you'll learn is known as, I believe, transduction. Okay, where the virus has the DNA and it just incorporates into the bacteria. Okay, so all they did was for transduction. They took a virus, put it in bacteria, allowed the virus to incorporate its genes into the bacteria. Okay, which were used to make pathogen-specific antibodies. Okay. So the bacteria produced these viral proteins from the new DNA from the virus, and that made antibodies. Okay, and these antibodies, your own body produces them too. Okay, they go out searching for antigens on foreign cells. Antigens are just proteins on cell receptors, and when that antibody detects that foreign body or that foreign cell, it's going to elicit an immunological response. It's going to activate all the other lymphocytes, the granulocytes, to come in and attack that foreign body. Okay. These antibodies bound to capillary walls in patient lung tissues. Okay, so these antibodies are going and attacking the capillaries of your lung tissue, meaning the alveoli, or the capillaries surrounding the alveoli of your lung tissue. Now, your capillary is it's basically the smallest vessels you have in your circulation, okay? That's where the diffusion of gases, oxygen gas and CO2 occurs at the side of your lungs. So all the blood goes through your capillaries and will basically, the gases, oxygen will diffuse from the metabolic waste, carbon dioxide, will diffuse from the blood in the capillaries straight into that lung tissue, okay? So if there's some pathogen in that lung tissue, it's probably making it very difficult for the gas to go from the capillaries to diffuse into the lung tissue. That's why these patients are having so much trouble breathing. Okay. Anything? I mean, now what do you guys think? Is that it? So, does this passage look? Do you think it's going to be easy? Do you think it's going to be difficult? Easy, difficult, easy, easy. This is going to be an easy passage because we've dissected everything. Okay. So so question 112. Which of the following statements about pulmonary function best describes all of the results graphed in Figure 1? Okay, we've already talked about this. They deteriorated in the first couple of days, were treated, probably got rid of the pathogen in the lungs, probably destroyed that virus in the lungs, and they all recovered according to that graph, right? So we have to use a process of elimination. It was less than 1/2 normal for at least a week. Okay, so they said the normal lung function was between 400 and 450. So does that make sense? It was less than 1/2 for at least a week. Yeah, I guess it was less than 1/2 for most of it. Well,
it was like 5 days.
It was like 5 days, right? Yeah, because the first 3 days they were all good, right? So I'd say by day 3 they were all kind of struggling, and so if you look at the first person, he got half his function by day 3. He lost half function by day 3. He recovered half his function by day 7. So that's not a week, right? So that's not the best answer. It declined 4 days after the onset of the illness. Okay. So the onset of the illness was pretty quick, within a day. Now, did they recover within 4 days after that? 1, 2, 3, 4. No, they didn't really decline 4 days after. They pretty much recovered after the onset, right? The onset was only 1 day because they deteriorated so rapidly, right? So after they deteriorated in that 1 day, it took them, 1, 2, 3, 4. They didn't really decline. Like they declined for maybe 1 or 2 days, 1, 2, and then recovered after that, right? So it didn't take 4 days, right? So I would not say that's the best answer. And remember, the MCAT is always about finding the best answer. Okay, a lot of the times there's 2 good answers. You've got to find the best answer. Okay. Okay, so answer B is not the best answer. C, it showed a rapid decline followed by slower recovery. And that makes sense. In the very beginning of the graph, they declined very rapidly, and then they recovered pretty slowly, right? That's a pretty good answer. D, it showed a slow decline followed by, no, that's wrong, right? Answer C. Okay. 113, what was contained in the sera from the respiratory patients of experiment 1 that caused this sera to react to the hantavirus that causes kidney disease? Okay, what was contained in the sera from the respiratory patient that caused the sera to react with the hantavirus that causes DNA, that causes kidney disease? Okay, well, thinking back to immunological response, you will get an immunological response when your body's immune cells detect a foreign body, that it recognizes as a foreign, something that's going to harm the body. Now, most of the time, your immune system has to train itself, and you'll learn this throughout the course. Okay, and that's basically the development of memory cells. Okay, which they'll stay in your body. These are B lymphocytes that will stay in your body, looking for, foreign bodies that it's detected years ago, in the past. Okay, so basically, what this says is, well, if there's a hantavirus that causes kidney disease, now there's no kidney disorder here. Okay, this is a lung disorder. But the hantavirus